Hello, my name is Amanda Savoie. I'm a research scientist who studies seaweed, and I'm director of the museum's Center for Arctic Knowledge and Exploration. Today, I'm going to tell you about the museum's Arctic seaweed collections and why they're important. Seaweeds, lichens, moss, and vascular plant collections are stored in what is called a herbarium. At the museum, we maintain the National Herbarium of Canada, where specimens from all across the world are stored in perpetuity for all Canadians. The herbarium is like a library filled with flat plants. When plants are collected in the field, they're pressed and dried so that they can be preserved. Next, they're mounted onto a sheet of special herbarium paper, and collection data is carefully noted. Finally, these sheets are carefully organized and then stored in cabinets. This works really well for vascular plants, but lichens and mosses are mostly stored in packets or envelopes because flattening them wouldn't work. But what about algae? Well, it depends on what kind of algae you're talking about. The algae collection can be split into two main groups, microalgae and macroalgae. The term algae does not refer to a single evolutionary lineage, but rather a group of more or less related organisms that are united by ecological traits. Algae are usually photosynthetic and usually aquatic, but there's a truly spectacular amount of diversity to be found in this group. Microalgae are tiny organisms that can only be seen using a microscope and include groups like diatoms, dinoflagellates, and desmids. These beautiful organisms can be difficult to study and collect because of their tiny size. They're stored in the collection in carefully curated and organized vial. A single vial can include many specimens and many species. In comparison, macroalgae are much larger and can be seen with the naked eye. They can be found in freshwater, but are much more likely to be marine. Seaweed is the colloquial term for marine macroalgae, and seaweed diversity is the focus of my research program. There are three main groups of seaweeds: red, green, and brown algae. They're found in all shapes and sizes, from tiny filamentous species to giant kelps more than 40 meters in height. So how are these messy seaweeds stored in the herbarium? Seaweeds are pressed, just like land plants, except it takes them longer to dry, and they often stick themselves to the paper. Once collected, they are carefully arranged and then pressed in just the same way. And when they're dry, they can last for hundreds of years. The museum has thousands of seaweed and other algae collections in the National Herbarium of Canada, and many of these are from the Canadian Arctic. Arctic and northern collections are especially important because the Arctic is currently one of the most vulnerable environments on Earth. The importance of the Arctic is a theme that will be repeated throughout the talks today, but one of the key reasons that this area is so important is that the rate of climate change in the Arctic is 2 to 3 times the global average. Furthermore, Arctic coastlines are rapidly changing as ice melts and glaciers recede. Coastlines are some of the most threatened habitats in the world, and marine organisms are particularly sensitive to temperature shifts. All of these changes have an impact on seaweeds. Each Arctic collection represents a specimen that was found at a particular place in a particular time. Some of these places have changed beyond recognition in the last 100 years, and these specimens provide an invaluable record of this change. Arctic seaweeds at the museum mostly come from two important collecting efforts: the Canadian Arctic Expedition from 1913 to 1918, and a series of expeditions led by museum phycologist R.K.S. Lee between 1967 and 1976. Collections from the Canadian Arctic Expedition are over 100 years old and can be used to study how seaweed distributions have changed over time. Specimens were collected from the Western Arctic and Hudson's Bay, and 3 new Arctic species were described. 50 years later, Lee was the first phycologist to conduct a thorough survey of the Canadian Arctic, collecting specimens from as far as 82 degrees north. In all, he sampled from 105 sites across the Arctic, scuba diving under the ice, sometimes in a wetsuit. Lee also wrote a book about his collections called A Catalogue of the Marine Algae of the Canadian Arctic. He lists the species that he collected and reported 37 new records. 40 years later, this is still the only book that comprehensively lists seaweed species from the Canadian Arctic. These Arctic collections from 50 years ago and 100 years ago represent a snapshot in time of Arctic biodiversity from a hard-to-access environment. They can be used to study changes that occurred in the past and to help model our predictions for the future. I hope I've convinced you of the importance of Arctic collections and flat seaweeds and inspired you to visit the herbarium sometime. Thank you for listening, and enjoy the rest of the talks.